In 1957, a 15-year-old boy found a fossil in a rock that wasn't supposed to have one. The rock was in Charnwood Forest, England. Geologists had already decided it was too old, formed long before anything complex could possibly exist. Life that organized, that structured, simply hadn't evolved yet. That was settled science. Except there it was, pressed into stone, over half a meter long, in a precise repeating pattern, formed roughly 575 million years ago. Tens of millions of years before the first animals. Tens of millions of years before anything was supposed to be that complex. It had no mouth, no gut, no eyes, no brain, and it was not alone. Fossils just like it, or nothing like it depending on which expert you ask, turned up on every continent, across every ocean basin, all within the same narrow window of time. Some were the size of a human body, some spread across the ancient seafloor in shapes that don't match any living thing we've ever found, before or since. They left impressions in rock, not bones, outlines, not organs, shapes, but no clear story. Scientists have been arguing about what they were for nearly 70 years. They are not plants, not fungi, not animals. One serious proposal is that they belong to a category of life that simply no longer exists, something that lived, thrived across the entire planet, and then disappeared so completely that we don't even have a name for what it was. And then they were gone, not slowly replaced, not clearly outcompeted. The record just ends before the first shell, before the first claw, before anything with a hard body left a trace of what it was hunting, something that dominated this planet on a global scale for millions of years, vanished, and we still don't fully know why. What were they? How did they live? And what actually happened to them? Today we examine Earth's first complex life and follow the evidence left in stone and the limits of what that evidence can still reveal. But before we start, let me know in the comments if you have any topic suggestions you would like me to cover in future videos. Now, settle in and let's go back to before the world had eyes and try to understand what was already living in it. Before anything recognizable could take shape, the planet moved through an expanse of time so long it resists comparison. Earth formed 4.54 billion years ago, a molten surface slowly cooling beneath a dim young sun. By around 3.7 billion years ago, life had already begun, not as creatures, but as chemistry learning to persist. These earliest forms left no silhouettes, no impressions of bodies, only faint chemical traces in ancient rock. For the next 3 billion years, that simplicity held. Nothing grew beyond the scale of a single cell. No limbs reached into water. No structures rose above the surface of the seafloor. The entire living world existed as microscopic forms, dividing, drifting, and enduring. This stretch of time is longer than the age of the solar system as measured from the present day. It is not a pause, but a steady, unbroken continuity of smallness, an immense patience written into the fabric of the planet. Across shallow marine environments, one form came to define this era. Stromatolites built themselves slowly, layer by layer, as communities of cyanobacteria trapped sediment and bound it into stone. These structures rose in low domes and ridges, spreading across coastlines and tidal flats. They still exist today in places like Shark Bay, where the water remains inhospitable to most other life. Their form has changed almost not at all across 3.5 billion years, a continuity so complete it becomes difficult to separate past from present. Within these layered communities, a quiet transformation began. Cyanobacteria used sunlight to split water molecules, releasing oxygen as a byproduct. At first, this oxygen vanished into the ocean, reacting with dissolved iron and other elements. Over time, those sinks filled. Around 2.4 billion years ago, oxygen began to accumulate in the atmosphere itself. For the organisms that had evolved in its absence, this was a toxin, corrosive and lethal. Entire lineages declined or retreated into isolated environments where oxygen could not reach them, yet the atmosphere was changing, molecule by molecule, toward a state that would eventually support more complex forms. Oxygen allowed new kinds of metabolism, more energetic, more demanding. The conditions required for larger bodies were assembling, though no such bodies yet existed. Much later, the planet entered a sequence of extremes. Between roughly 720 and 635 million years ago, ice advanced across the surface of the oceans, reaching even equatorial regions. The world passed through multiple intervals where glaciers dominated the seas, sealing vast stretches beneath frozen cover. These were not singular events, but cycles, advancing and retreating over millions of years. Each thaw altered the chemistry of the oceans. As ice withdrew, nutrients locked in rock and sediment washed into the water, enriching it. Shallow seas warmed again, circulation resumed, and the conditions shifted. These cycles did not produce immediate complexity, but they reshaped the environment repeatedly, opening and closing possibilities. When the final glacial period ended, around 635 million years ago, the stage was different. Oxygen had accumulated, the oceans carried new chemical balances, and ecological space existed in forms that had not been available before. Within roughly 60 million years, the seafloor would no longer resemble the quiet microbial world that had persisted for billions. The timescale, the oxygen, and the ice remain beneath everything that follows, not as background, but as the slow construction of a world that could finally begin to change. A shape on stone can pass unnoticed for centuries, waiting for a moment when someone decides to look a little longer than expected. In the summer of 1957, that moment arrived in a quiet stretch of woodland in Charnwood Forest, where exposed rock surfaces carried the weight of deep time without drawing attention to themselves. A school group moved across the outcrop, their attention scattered between instruction and curiosity. Among them was Roger Mason, 15 years old, pausing at a cliff face, where the stone held a pattern that did not quite belong to fracture or mineral vein. It spread outward in a repeated arrangement, branching with a precision that suggested growth rather than accident. He photographed it, an act simple enough, then carried the image back to his teacher, and from there it began a quiet passage through academic hands until it reached Cambridge. The rock that held the impression had already been given a place in the timeline. It was labeled Precambrian, part of an age understood to precede any complex life. That understanding rested on decades of work, shaped in part by figures like Charles Walcott, whose interpretations helped define where complexity was allowed to begin. According to that framework, anything organized beyond the simplest forms could not exist in stone of that age. The pattern Mason had noticed did not fit within that boundary. The fossil was eventually named Charnia masoni, its structure described as a frond, anchored by a holdfast, rising from the ancient seafloor into water that no longer exists. It could reach lengths of 2 meters, its branches arranged in alternating sequences along a central axis, each segment echoing the one before it. The symmetry was not decorative, but systematic, a record of growth written into rock. Yet this moment of recognition was not the beginning. More than a decade earlier, in 1946, Reginald Sprigg had walked through the arid landscape of the Ediacaran Hills and found impressions pressed into red sandstone, discs and fronds preserved in silence. He understood their significance well enough to send his findings to publication. The response did not match the discovery. His work was rejected, then presented in person and set aside again, not through lack of evidence, but through conflict with expectation. The prevailing view held firm. If the theory did not allow for such organisms, then the impressions were interpreted as something else: inorganic structures, sedimentary features, anything that would preserve the established timeline. Spriggle's fossils remained where they had been placed, acknowledged in fragments, but not integrated into the larger picture. Over time, attention moved elsewhere, and he followed a different path beyond academic geology. Only later, when Mason's discovery forced a second look, did the earlier evidence return to the surface with new context. The scattered pieces began to align, not through a single moment of realization, but through gradual acceptance that the record had been read too narrowly. The organisms from South Australia and England belonged to the same interval, one that had not yet been formally recognized. The name given to that interval, the Ediacaran, comes from those hills where Sprigg first paused among the stones. It was not officially defined until 2004, making it the most recent addition to the geological timescale, a period that had existed in the rock all along, waiting for the framework to expand enough to include it. In this sequence of discovery and delay, there is a quiet pattern. Evidence arrives without announcing its place, and understanding follows only when the boundaries around it begin to shift. If the stone is read not as surface, but as presence, each impression becomes less like a fossil and more like a brief encounter, something that existed fully and then withdrew, leaving only the outline of its occupation behind. Dickinsonia appears first as a flattened oval, its body divided by a central seam, with repeated segments extending outward in quiet symmetry. The surface carries a softness, even in rock, as if it once rested directly against the ground beneath it. It could reach 1.4 meters in length, though it begins at scales barely visible. No opening suggests ingestion, no structure implies internal processing. It moved by contact alone, sliding across the surface and drawing nutrients through its entire underside. Where it paused, the substrate beneath it was altered, a subtle absence marking where the organism had fed before shifting forward again. Nearby, Charnia rises upward, anchored at a single point. Its form unfolds in alternating branches along a central axis, each segment repeating the previous at a slightly smaller scale, creating a pattern that extends both outward and inward at once. It did not lie against the surface, but extended into the water column, growing in environments untouched by light. Its structure carries a kind of order that feels deliberate, though no comparable living form offers a clear parallel. Set into the same ancient surfaces, Trabrachidium forms a low dome, its surface marked by three curved arms spiraling from a central point. The pattern rotates rather than mirrors, a symmetry that does not align with the bilateral forms that dominate later life. It suggests movement without direction, a geometry that does not resolve into front or back, only a continuous circulation across its surface. In contrast, Cambrella introduces a different presence. Its body shows a distinct leading edge, a boundary that consistently faces the direction of movement. Around it, fine marks spread outward in arcs, the result of repeated scraping against the surface. These traces indicate action, a deliberate interaction with its surroundings, as if the organism could locate and process specific areas rather than absorbing passively. Sprishana remains smaller, its form divided into segments with one end broader and more defined. The outline hints at a directional body plan, though its exact relationship to later segmented animals remains uncertain. It carries the suggestion of familiarity without resolving into anything fully recognizable. Across these surfaces, Fractifus extends in elongated shapes, its presence repeating in clusters that appear connected. New individuals formed along horizontal extensions, spreading outward across the substrate in a pattern that resembles growth rather than dispersal, as if the organism expanded by reaching into adjacent space. Partially embedded within the sediment, Pteridinium presents a structure divided into three flanges, each composed of interlocking units arranged in a fan. Its body occupies both the surface and the layer beneath, suggesting a mode of life that bridged two environments simultaneously. Then there is Io Andromeda, its form spreading outward in 8 radiating arms, each branching further as it extends. The pattern resembles a spiral expanding into space, ordered yet unfamiliar, without clear relation to any modern lineage. These organisms are not isolated occurrences. More than 200 distinct species have been identified across every continent, including Antarctica, their forms preserved in separate regions yet confined to the same interval of time. The distribution is global, the diversity extensive, and the coherence unmistakable. It was not a scattered experiment, but a complete world, composed of bodies that followed rules no longer present in the life that came after. What lies beneath them is not empty space, but a living surface, continuous and quietly active, shaping every form that rests upon it. The ground itself is part of the organismal world, not separate from it, and without it, the bodies impressed into stone lose their meaning. Across vast stretches of the ancient seafloor, a thin but persistent layer spread outward in all directions. This was the mat ground, a dense, cohesive sheet formed by bacteria and cyanobacteria, bound together into a structure that held both sediment and chemistry in place. It was not a loose accumulation, but a unified surface, centimeters thick, stretching across shallow marine environments with a consistency that replaced the shifting sands of later oceans. Beneath this layer, the sediment remained undisturbed. There were no burrows cutting through it, no tunnels or cavities interrupting its structure. Each layer settled in sequence, forming a delicate stratification that preserved the chemical gradients within it. Oxygen and other compounds remained confined to specific depths, creating a stable arrangement that persisted over long intervals. Above it, the mat sealed the surface, holding everything in quiet continuity. For the organisms that appeared later, this surface was both foundation and resource. Forms like Charnia anchored themselves directly into it, their holdfasts embedded within the living layer. Others, such as Dickinsonia, moved across it, maintaining constant contact with the mat beneath them. The boundary between organism and environment remained thin, almost indistinct, as if the two were part of the same extended system. Within the mat, organic material accumulated and broke down, releasing dissolved compounds into the surrounding water and the surface itself. This created a continuous field of available nutrients, not concentrated in discrete sources, but distributed across the entire substrate. Many Ediacaran organisms appear to have relied on this condition, absorbing these molecules directly through their outer surfaces. This mode of feeding, known as osmotrophy, required no specialized structures, only proximity and contact. The result was a world without urgency. Growth occurred slowly, without interruption or competition for fleeting resources. There is no sign of pursuit, no indication of one organism overtaking another. The absence of disturbance is visible in the fossil record itself. Across tens of millions of years, there are no repaired injuries, no hardened exteriors, no adaptations that suggest defense or escape. The organisms appear as they were, unaltered by conflict. Paleontologists have described this setting as a kind of garden, not in appearance, but in its ecological character. A stable surface, consistent conditions, and organisms arranged across it without evidence of disruption. The term reflects an environment where interaction did not involve damage, where presence did not require resistance. When these organisms died, they often remained in place. The mat continued to grow, covering their forms with a new layer before decomposition could erase them. This process preserved their outlines with remarkable fidelity, sealing them within the sediment below. The impressions that remain are not just shapes, but records of a moment held still by the very surface that supported it. Everything about these organisms is tied to this substrate. Their structure, their feeding, their position in the environment all depend on the conditions the mat ground maintained. It was not simply the surface they occupied, but the system that made their existence possible. Inside each living cell, energy moves quietly, carried through reactions too small to see yet decisive in what a body can become. The scale of life is limited not only by structure, but by how much energy can be produced, sustained, and distributed across that structure. For most of Earth's history, that energy remained constrained, and life stayed correspondingly small. To build a large body requires a different kind of metabolism, one that uses oxygen to release far greater amounts of energy from the same chemical sources. This process, aerobic respiration, supports growth, maintenance, and coordination across many cells at once. Without sufficient oxygen in the surrounding environment, such complexity cannot be maintained. The atmosphere, for much of early Earth, did not provide that condition in a stable or widespread way. Estimates suggest that organisms resembling animals require oxygen levels reaching at least a few percent of what exists today, perhaps around 3% of modern atmospheric concentration. Below that threshold, energy budgets remain too limited to sustain large, organized forms. By the time the Ediacaran world emerged, the planet appears to have crossed that boundary, not briefly, but consistently, enough to support new possibilities. Yet oxygen alone does not account for the long delay. Between roughly 1.8 billion and 800 million years ago, life entered a prolonged interval where change slowed almost to stillness. This span, sometimes called the boring billion, saw stable ocean chemistry and relatively constant atmospheric conditions. Microbial life persisted and diversified at small scales, but no major shift toward complexity took hold. The reasons remain uncertain, as if the conditions for change were present but not yet aligned. Another requirement lies deeper, within the structure of the cell itself. Complex multicellular organisms are built from eukaryotic cells, which contain a nucleus and internal compartments that organize different functions. These cells did not arise gradually through a series of small modifications. Instead, they appear to have originated from a single, rare event in which one microbe entered into a lasting partnership within another, eventually becoming the mitochondrion. This merger provided a new level of internal energy processing, allowing cells to support larger genomes and more intricate regulation. Even with this foundation, the genetic instructions for building a body seem to have appeared long before they were expressed in visible form. Genes that control development, including those that determine spatial organization within an organism, were already present in single-celled ancestors. The capacity to generate complex structure existed but remained unused, as if waiting for conditions that would allow it to unfold. The environmental cycles that reshaped the planet also played a role in this timing. Repeated global glaciations disrupted established ecosystems, clearing ecological space that had been occupied for immense spans of time. When the ice retreated, new conditions emerged, with altered nutrient availability and reorganized habitats. These transitions did not create complexity directly, but they removed constraints that had limited its expression. What emerges from this sequence is not a single cause, but a convergence. Oxygen levels reached a usable threshold. Cellular architecture provided the necessary internal organization. Genetic systems, capable of directing growth, were already in place. An environmental change opened space for new forms to persist. Around 575 million years ago, these elements aligned in a way they had not before, allowing life to extend beyond its long confinement into new scales of form. Among all the impressions left behind, one form appears again and again, as if the stone itself kept returning to the same shape, repeating it across distant places and quiet intervals of time. Dickinsonia lies low against the surface, its body broad and flattened, divided by a central furrow that runs from one end to the other. From this axis, segments extend outward in paired rows, each one gently offset from its neighbor, forming a pattern that feels both ordered and unfamiliar. The resemblance to a leaf or a quilted surface arises naturally, though neither comparison fully contains it. Its growth followed a direction that can be read along its length. New segments appeared at one end, added in sequence, while the older portions remained in place, gradually becoming proportionally smaller as the organism expanded. This process, known as terminal addition, creates a body that records its own history, each segment marking a moment of development. Similar strategies exist in some modern organisms, yet none reproduce this exact arrangement, leaving Dickinsonia without a clear counterpart. The surface beneath certain specimens carries a second kind of record. Adjacent to the outline of the body, there are areas where the underlying layer was altered, a subtle clearing that matches the shape of the organism. In some cases, multiple such impressions appear in sequence, each one slightly offset from the last. The pattern suggests movement, not through limbs or appendages, but through the entire underside of the body, making contact, feeding, and then shifting forward. The organism did not pass quickly, but progressed in a slow, deliberate series of placements. Its size varied widely. The smallest forms are barely visible without magnification, while the largest extend to 1.4 meters across, making them among the most expansive organisms of their time. Despite this range, the structure remains consistent, scaled rather than altered, as if governed by a fixed set of developmental rules. In 2018, a new layer of evidence emerged from specimens found along the White Sea coast of Russia. Jochen Brox and his team at Australian National University analyzed organic molecules preserved within the fossilized remains. They identified cholesterol derivatives, compounds associated with the cell membranes of animals. This finding marked the first time a direct biochemical signal had been linked to an organism from this period, suggesting that Dickinsonia may belong within the animal lineage. The presence of cholesterol carries weight, as it is not produced in the same form by bacteria or fungi. It implies a cellular structure aligned with animal biology, even if the external form does not resemble any known animal. The discovery moved the discussion beyond shape and into chemistry, offering a different kind of classification. Yet the interpretation is not without uncertainty. Organic molecules can migrate through rock over long spans of time, and contamination from surrounding material remains a possibility. The conditions at the White Sea site are unusual, preserving details not seen elsewhere, which raises further questions about how representative the signal may be. Some suggest that the detected compounds could originate from organisms that interacted with the fossil after its formation. Even if the cholesterol is accepted as original, the anatomy of Dickinsonia remains unresolved. No structures corresponding to a digestive system, nervous network, or reproductive organs have been identified. Its body does not align with known animal architectures, lacking the features typically used to define them. It would be an animal in composition, yet constructed along principles that diverge from those seen in later forms. In this way, Dickinsonia sits at the center of the broader uncertainty. It connects chemical evidence to a form that resists classification, bridging categories without fully belonging to any of them. Not all traces of life are bodies. Some remain as marks of action, patterns that record what an organism did rather than what it was. In a series of ancient surfaces along the White Sea, these marks appear as delicate fans of fine lines, spreading outward in repeated arcs, each set anchored to a single point where something once rested. At the center of these patterns lies Cambrian, an oval form with a distinct boundary between its outer edge and interior. Its body could reach up to 15 centimeters in length, with a flexible central region enclosed by a more rigid rim. Unlike many of the organisms that share its environment, Cambrian shows a consistent orientation. One end leads, the other follows, a subtle asymmetry that becomes visible when its position is compared across multiple specimens. The surrounding marks reveal more than its outline. Each fan consists of parallel grooves arranged closely together and repeated in sequences that extend outward from the organism's position. These are not random disturbances, but structured traces produced by a repeated motion applied to the surface. The most widely accepted interpretation is that Cambrian possessed a rasping structure, an organ capable of scraping material from the substrate. With each movement, it removed a thin layer, leaving behind the fine, radiating patterns now preserved in stone. This mode of feeding represents a shift from passive absorption to active acquisition. Instead of relying on dissolved compounds available at the surface, Cambrian appears to have sought out and processed specific areas, moving from one location to another as resources were depleted. The pattern of marks suggests a sequence of actions, a progression across the surface guided by more than immediate contact. Such movement implies coordination. To travel in a consistent direction and engage with the substrate in a repeated, purposeful way requires some form of internal signaling. Even in its simplest form, this would involve the detection of environmental cues, the processing of those cues, and the execution of movement in response. The structures responsible for this would have been minimal compared to later systems, yet their presence marks a step toward integrated behavior. Because of these features, some researchers have proposed that Cambrian may be related to early mollusks. Its combination of a flexible body, a firm outer margin, and a rasping feeding mechanism resembles, in a distant way, the organizations seen in modern snails and their relatives. If this connection holds, Cambrian would represent an early stage in a lineage that continues into the present, linking these ancient forms to a wide range of living organisms. The interpretation remains debated. The grooves surrounding Cambrian could have been produced by another organism interacting with the same surface, and the similarities to mollusks may reflect convergence rather than shared ancestry. The fossil record does not preserve the soft internal structures that would resolve this directly, leaving the relationship open to multiple readings. Even within that uncertainty, one aspect remains clear. Cambrian demonstrates that complex behavior was already present during this interval. It moved with direction, interacted with its environment in a repeated and structured way, and left behind evidence of those actions. In doing so, it adds a new dimension to the record, one that cannot be seen in form alone, but must be read in the patterns of activity preserved alongside it. Along a remote coastline in Newfoundland, the rock surface tilts toward the sea, exposing plains that were once horizontal, once resting quietly at the bottom of an ancient ocean. Here, within the boundaries of Mistaken Point Ecological Reserve, entire communities remain fixed in place, not scattered or rearranged, but preserved as they existed in a single moment. This assemblage represents the earliest known gathering of complex organisms, dating between roughly 575 and 560 million years ago. The surfaces are marked by thousands of impressions, each one recording the position of an individual within a living landscape. The arrangement is not random. It reflects spacing, orientation, and repetition, a structured environment held intact across immense time. The preservation of these communities occurred through sudden burial. Volcanic ash, settling through the water column, descended onto the seafloor and covered everything in a fine, even layer. The organisms beneath were sealed in place, their forms pressed into the sediment before decay could erase them. What remains is not a collection assembled over time, but a snapshot, a complete surface captured in a single event. Geochemical analysis of the surrounding rock reveals a condition that shapes how these organisms are understood. The depth at which they lived lay well below the reach of sunlight. No photosynthesis could take place here, no energy drawn from light. Yet the organisms present are among the most structurally intricate of their time. This absence of light removes one of the most common pathways for life, leaving only chemical sources available. Within this environment, a group known as rangeomorphs dominates the assemblage. Their bodies consist of branching elements that repeat the same pattern at multiple scales, from the smallest segments to the full extent of the organism. Each branch divides into smaller branches, which divide again, maintaining the same geometric structure throughout. This pattern, called self-similarity, creates a form that expands outward while preserving its internal organization. The geometry serves a purpose. By repeating the same structure at different scales, these organisms maximize the surface area available relative to their volume. In an environment where nutrients exist in low concentrations, dispersed through the surrounding water, this increased surface provides more interface through which dissolved compounds can be absorbed. The body becomes a network designed for exchange, extending into the water column to intercept what passes through. The arrangement of these organisms across the surface reveals a further layer of organization. Some remain close to the substrate, their forms spreading outward along the ground. Others rise higher, extending upward into the water. The result is a vertical distribution, a separation of space that allows multiple forms to occupy the same area without direct overlap. It resembles a layered structure, where different heights correspond to different modes of existence within the same environment. Across single surfaces, multiple size classes of the same organism appear together. Smaller individuals occupy the same space as larger ones, indicating ongoing growth and the presence of successive generations. These were not isolated occurrences, but stable populations, maintaining themselves over time through continuous reproduction. What emerges from this site is a picture of early complexity that does not begin in shallow, illuminated environments, but in depth and darkness. The organisms here developed intricate structures without reliance on light, drawing instead on the chemistry of the surrounding water. Their forms reflect optimization rather than experimentation, as if they arrived already adapted to the conditions they inhabited. In this preserved landscape, the earliest complex life appears not as a tentative beginning, but as an established system, complete in its structure and quietly sustained in a world without illumination. Where the shoreline of White Sea meets exposed stone, layers of sediment open like pages, each one holding a separate interval of a world no longer present. Along this coast, Within the Vendian deposits of the Arkhangelsk region, the record does not compress into a single moment. It unfolds across time, preserving a sequence rather than a snapshot. Decades of work, led by Mikhail Fedonkin and his collaborators, have revealed a remarkable diversity within these layers. More than 40 distinct species have been identified, represented by thousands of individual specimens distributed through multiple horizons. Each layer corresponds to a different interval, together spanning roughly 10 million years. Instead of a single preserved surface, this site offers a continuous record of change. The conditions of preservation vary across these layers, but often involve rapid burial by fine sediment carried in by storms. As currents shifted and silt settled, organisms were covered gently yet completely, capturing details that are rarely retained elsewhere. Surface textures, edges, and subtle variations in form remain visible, allowing growth stages to be distinguished. Individuals of different sizes appear within the same species, providing a sense of development through time. Some of the most striking specimens remain in their original positions. Frond-like organisms are found still attached at their base, their structures collapsed but not transported. This confirms that they grew upward from the seafloor, extending into the surrounding water rather than lying across the surface. The orientation of these forms, preserved without displacement, offers a direct view of how they existed in life. Because the sequence extends across millions of years, it becomes possible to observe patterns of appearance and disappearance. Certain species emerge in lower layers, persist through several intervals, and then no longer appear in the higher strata. Others are confined to narrower ranges, existing briefly before vanishing from the record. This layered arrangement allows the timing of these changes to be traced with a level of resolution not available at other sites. Among the organisms found here are forms not seen elsewhere, and Diva displays a bilateral structure that stands apart from other known examples, suggesting a different organizational pattern. Ventogyrus appears as a conical body with internal compartments, indicating a three-dimensional arrangement that may have extended partly beneath the surface during life. Io Andromeda returns here as well, its radiating arms preserved with clarity that reveals the consistency of its form across different locations. The arrangement of organisms across individual surfaces is not random. Certain species tend to appear together, forming clusters that repeat across multiple layers. Others are more isolated, occupying spaces where different conditions may have prevailed. These patterns suggest interactions that are not directly visible, possibly related to competition for space or subtle chemical influences within the environment. As the sequence approaches its uppermost layers, a shift becomes apparent. The assemblage that dominates the earlier intervals begins to thin. Some species no longer appear, replaced by different forms that occupy the same general space. This transition occurs between approximately 558 and 555 million years ago, marking a change that is gradual rather than abrupt. In this extended record, the White Sea preserves not only what existed, but how it changed, offering a rare view of continuity within a world that would not remain as it was. Some of the most revealing records are not bodies at all, but movements translated into stone. These traces remain where actions once unfolded, preserving direction, repetition, and change without preserving the organism itself. They appear as lines, grooves, and subtle disruptions, each one a fragment of behavior held in place long after the body that made it is gone. The earliest of these markings are simple. Shallow paths extend across ancient surfaces, faint depressions left by organisms moving in contact with the ground. These trails do not cut into the layers below, but rest upon them, tracing a route rather than reshaping the environment. The motion they record is basic, a steady progression across the surface, guided by proximity rather than depth. Over time, these traces begin to vary. Patterns emerge that suggest repeated actions, not just movement, but interaction. Feeding marks appear alongside pathways, indicating that some organisms did more than pass across the surface. They engaged with it, leaving behind evidence of contact that altered the immediate environment in localized ways. The record does not change abruptly, but accumulates, each new pattern adding to the range of behaviors preserved. Around 555 million years ago, a different kind of mark begins to appear. Instead of extending horizontally, these traces descend. Narrow openings penetrate the surface and continue downward into the layers below. This shift from surface movement to vertical excavation marks a change not only in behavior, but in the structure of the ecosystem itself. To move through sediment in this way requires a different set of biological capabilities. The organism must process material as it advances, taking it in at one end and releasing it at another. This implies a continuous internal passage, a through gut, allowing sediment to pass through the body. Coordinated movement is necessary to displace material and maintain direction, while some form of sensory awareness must guide the organism through an environment it cannot see. As these burrows increase in number and complexity, they begin to alter the ground itself. Sediment that once remained layered and stable becomes mixed. The distinct boundaries between layers blur as material is moved and reworked. Oxygen, which had been confined to upper regions, penetrates deeper, changing the chemical conditions below the surface. The structure that had persisted for immense spans of time begins to shift. This process, known as bioturbation, transforms the seafloor from a stable surface into a dynamic environment. The continuity that once defined it is broken, replaced by a system in which the ground is constantly being reshaped by the organisms moving through it. The earlier condition, preserved in undisturbed layers, gives way to a new state where such preservation becomes increasingly rare. As burrowing continues, the surface layer that supported earlier forms becomes fragmented. The cohesion of that layer depends on its continuity, and once it is disrupted from below, it cannot maintain itself. The rate at which it is broken apart exceeds the rate at which it can reform. The ground that once provided a stable interface begins to lose its integrity. The progression of these traces reflects a broader transition. In the final 15 million years of this interval, the increasing complexity of burrows corresponds with a reduction in the diversity of earlier body forms. The patterns left behind by one group of organisms record the expansion of their activity, while the absence of others marks their decline. In this way, the record of behavior becomes a measure of change, showing not only what organisms did, but how those actions reshaped the conditions in which life continued. Classification depends on boundaries, on the ability to place an organism within a structure that defines what it is and how it relates to others. In this interval, those boundaries begin to dissolve. The forms preserved in stone resist placement, not because they lack detail, but because the details do not align with the categories built from later life. One interpretation places some of these organisms within the animal lineage. Evidence drawn from molecular analysis, such as that associated with Dickinsonia, suggests a biochemical affinity with animals. Behavioral traces linked to Cambrian indicate movement and interaction consistent with early animal activity. In addition, simple sponge-like forms appear in deposits from this period, providing a possible connection between these ancient organisms and the earliest known animals. Under this view, At least part of the assemblage represents the beginnings of animal life. Another perspective draws comparisons with fungi. The flattened, spreading forms of some organisms resemble growth patterns seen in fungal networks, where expansion occurs across a surface rather than through differentiated structures. The absence of internal organs, combined with a mode of nutrient acquisition based on absorption across the body's surface, aligns with how fungi interact with their environment. From this angle, these organisms could represent a form of multicellular organization closer to fungal systems than to animals. A third proposal suggests a relationship with lichens. Certain frond-like structures bear a superficial resemblance to lichen growth, particularly in their branching patterns and attachment to substrate. This interpretation, however, encounters difficulty when applied to environments where light does not reach. Without access to photosynthesis, the defining partnership within lichens becomes impossible, limiting the scope of this explanation. In 1992, Adolf Seilacher introduced a different possibility. He proposed that many of these organisms belong to an entirely separate category of life, which he named Vendobionta. In this view, the organisms are not early animals or fungal analogues, but representatives of a distinct mode of multicellular existence that has no modern equivalent. Their bodies appear to be constructed from repeated units, arranged in geometric patterns that form a kind of quilted structure. This quilted organization suggests a method of support and distribution unlike that found in later organisms. Instead of relying on skeletons or circulatory systems, these bodies may have consisted of fluid-filled compartments arranged in interconnected networks. The geometry provides stability, while the internal compartments allow for the movement of nutrients without specialized vessels. The structure resembles an air-filled surface, where each section contributes to the integrity of the whole. A fifth interpretation steps away from the idea of a single classification. It proposes that the group commonly referred to as the Ediacaran biota may not represent a unified lineage at all. Instead, it may be a collection of organisms that share an environment rather than a common ancestry. Under this model, early animals, fungal-like forms, and entirely extinct lineages could coexist within the same ecological setting, each following different biological principles while appearing together in the fossil record. At present, many researchers lean toward this mixed origin. Some organisms show characteristics that align with known groups, while others remain entirely distinct. This approach accepts that no single framework can encompass all the forms present, though it leaves many individual species without a clear placement. The difficulty in resolving these interpretations arises from the nature of the evidence itself. Soft-bodied organisms preserve external outlines but rarely retain internal structures. Without access to anatomy, classification relies on shape and pattern alone. The result is a taxonomy built from impressions, where the essential features that define relationships are largely absent, and the organisms remain partially concealed within their own outlines. The record shifts not with a single boundary, but with a change in what becomes visible. At approximately 538.8 million years ago, a new interval begins, and within a span of roughly 20 million years, the forms preserved in stone expand into a range that includes nearly every major animal body plan known today. In the scale of Earth's history, this interval occupies less than half of 1%, a brief expansion following a much longer period of restraint. What appears in this interval differs in fundamental ways from what preceded it. Bodies now show defined fronts and backs, structures for sensing and interacting, appendages that extend outward into the surrounding medium. Hard parts begin to form, shells and protective coverings that resist decay and preserve more readily. Movement becomes varied, with organisms capable of pursuing, grasping, and responding to one another in ways not previously recorded. Among the most detailed records of this transition is found in the Burgess Shale, a deposit discovered in 1909 by Charles Walcott. The fine sediment in which these organisms were buried preserved not only their outlines, but also internal structures. Digestive systems, nervous pathways, and soft tissues remain visible, offering a level of detail that transforms how these early animals can be understood. Within this assemblage, forms appear that extend beyond familiar categories. Anomalocaris reaches lengths of up to 1 meter, with large compound eyes composed of thousands of individual lenses. At its front, circular mouthparts form a ring capable of processing prey, while jointed appendages extend outward to grasp and manipulate. Its structure reflects a level of coordination and sensory capacity not previously seen. Nearby, Opabinia presents a configuration that initially resisted interpretation. 5 eyes are arranged on stalks, providing a wide field of perception, while a flexible extension projects forward, ending in a grasping structure. When first described publicly, its appearance was met with uncertainty, as it did not align with established expectations of animal form. Hallucigenia offers another example of this unfamiliarity. Its body is supported by rigid projections on one side, while softer appendages extend from the opposite side. Early reconstructions placed it inverted, an arrangement later corrected as additional specimens clarified its orientation. The difficulty in interpreting its structure reflects how different these organisms are from later, more recognizable forms. One explanation for this rapid diversification centers on the emergence of image-forming vision. Once an organism could detect others visually, the environment changed. Visibility introduced new interactions, including avoidance and pursuit. Organisms capable of seeing could respond to threats or opportunities at a distance, and those unable to do so faced new pressures. This shift may have driven the development of additional features, including protective structures, increased mobility, and more complex sensory systems. The result is an interval where forms diversify quickly, filling ecological roles that did not previously exist. The variety of body plans expands, and with it, the range of interactions among organisms. The fossil record reflects this change through the presence of structures associated with feeding, movement, and perception, all appearing within a relatively narrow span of time. In comparing this interval to what came before, the difference is pronounced. The earlier forms, preserved as impressions without internal detail, give way to organisms with defined anatomy and varied behaviors. Whether these new forms emerged directly from earlier lineages or represent separate developments remains unresolved, but the transition marks a shift in the kinds of structures that persist in the record and in the ways life interacts with its environment. In the later layers of this interval, a new texture appears in the record, not as a pattern pressed into sediment, but as a structure that stands apart from it. Claudina forms narrow tubes composed of stacked cones, each segment fitting within the next, creating a continuous sheath. These tubes measure only a few millimeters across, yet they represent a shift in how bodies are built and preserved. The material of these tubes is mineralized, composed of calcium carbonate rather than soft tissue. This makes Claudina the earliest known organism to construct a rigid external structure. Instead of relying solely on form maintained by fluid or cellular cohesion, it produced a shell, investing energy into a barrier between itself and the surrounding environment. This development marks one of the most significant transitions within this period, as it introduces a new way of interacting with external conditions. The presence of such a structure implies a change in those conditions. Building a shell requires resources and metabolic effort, suggesting that the advantage gained from this investment outweighed its cost. A rigid exterior provides resistance, a means of protection that would not be necessary in a setting without disturbance. The appearance of this feature indicates that the environment had begun to include interactions that could damage or disrupt unprotected forms. Evidence of such interactions appears directly on the shells themselves. Specimens of Cloudina recovered from regions in what is now Namibia and China show small, circular perforations penetrating their walls. These openings are not random fractures, but consistent in shape and placement, indicating a repeated process. The pattern closely resembles the marks left by modern organisms that drill into shells to access the soft tissue within. This provides the earliest direct indication of predation involving penetration of a protective structure. The act of drilling suggests an organism capable of locating, attaching to, and processing another organism in a targeted way. The presence of these marks shows that such interactions were already occurring, altering the conditions under which life persisted. At roughly the same time, another organism appears with a similar departure from earlier forms. Namacalathus constructs a small, goblet-shaped structure, also composed of mineralized material. It attaches to the seafloor and extends upward, its form distinct from Cloudina, yet sharing the use of a rigid external framework. The near simultaneous emergence of these structures suggests that the development of mineralization was not isolated, but part of a broader shift. The reasons behind this shift are not fully resolved. One possibility is that the rise of organisms capable of damaging soft bodies created selective pressure, favoring those with protective coverings. Another is that changes in ocean chemistry made the formation of mineralized structures more accessible, reducing the cost of building them. Both factors may have contributed, interacting in ways that encouraged multiple lineages to adopt similar strategies. Cloudina and Namacalathus occupy a position at the boundary between two intervals, appearing near the end of one and continuing into the beginning of the next. Their presence bridges this transition, linking the earlier assemblages with the later expansion of organisms that rely extensively on hard parts. In the layers above, such structures become more common, forming the basis of a new kind of fossil record. The emergence of these forms indicates that interactions between organisms had changed in a fundamental way. The development of protective structures and the evidence of their penetration reflect a dynamic in which one organism's survival becomes directly tied to another's actions. This marks the beginning of a pattern that continues beyond this interval, shaping the diversity and complexity of the life that follows. In the desert of Namibia, the land opens in vertical faces of exposed rock, each layer marking a separate interval of time. These outcrops reveal ancient seafloors stacked one above another, allowing the passage of millions of years to be followed upward through stone. Here, within what is known as the Nama assemblage, the final phase of this interval is preserved between roughly 548 and 538 million years ago. The accessibility of these formations makes them distinct. Instead of isolated surfaces, the rock presents a continuous sequence where older layers lie beneath younger ones in clear progression. By moving along the cliff faces, it becomes possible to read the history of this world as a gradual shift rather than a single event. Each layer adds detail to a transition that unfolds over time. Among the organisms unique to this region is Rangia, a frond-like form that extends beyond simple surface impressions. Its internal structure is preserved in 3 dimensions, revealing compartments arranged with geometric precision. The body displays a threefold symmetry, dividing into repeated sections that interlock within the overall form. No direct relationship has been established between Rangia and any other known organism, leaving it as an isolated example of a design that did not persist. As the sequence is followed upward, a pattern begins to emerge. In the lower layers, a variety of soft-bodied forms appear, similar to those found in earlier intervals. Their diversity suggests a continuation of established conditions. Moving higher, these forms become less common. In their place, structures built from mineralized material begin to appear more frequently, including those associated with Cloudina and Namacalathus. In some layers, these organisms accumulate in dense clusters, forming structures that extend above the seafloor. These accumulations resemble early reefs, where multiple individuals contribute to a shared framework. The result is a 3-dimensional habitat, providing space and support for other organisms. This represents a shift from isolated individuals to collective structures that modify their environment. At the same time, the record of movement within the sediment becomes more pronounced. Burrows increase in depth and complexity, appearing more frequently as the sequence progresses. The changes observed in these traces reflect a gradual intensification of activity beneath the surface. What begins as simple penetration becomes a network of pathways extending through the sediment, altering its structure. This transformation is visible across the entire vertical sequence. Each successive layer records a slightly different state, with burrows becoming more intricate and widespread. The cumulative effect is a steady alteration of the environment, moving from a stable arrangement toward one that is continuously reworked. Near the uppermost layers, the earlier soft-bodied forms no longer appear. Their absence is not marked by a sudden boundary, but by a gradual thinning until they are no longer present. Above this level, in the overlying rock, the assemblage changes completely. The forms that dominated earlier layers do not continue into this new interval. What this sequence reveals is a transition extended across millions of years. The earlier world does not end abruptly, but recedes as new structures and behaviors become more prominent. The shift is continuous, each layer reflecting a slightly altered balance. In this vertical record, the passage from one state to another can be followed step by step as one system diminishes and another expands within the same space. Marks on shells appear as small circles, clean and deliberate, interrupting an otherwise continuous surface. These openings do not form through accident. They are placed, repeated, and aligned with the structure beneath them, suggesting an action carried out with consistency. From these marks, one interpretation extends outward, proposing that the disappearance of earlier forms may have been driven by the arrival of organisms capable of consuming them directly. The evidence begins with the perforations found in Cloudina, where the walls of mineralized tubes have been penetrated in a way that indicates access to the interior. The pattern of these openings resembles methods used by later organisms that drill through protective coverings to reach soft tissue. Alongside this, the increasing complexity of trace markings suggests a broader rise in active interaction, with organisms moving, engaging, and altering their surroundings in more direct ways. These changes occur at the same time that earlier forms begin to decline in number and diversity. The organisms that dominated earlier intervals appear, in this context, to be highly exposed. Their bodies lack rigid protection, relying instead on soft structure and direct contact with their environment. They show no indication of rapid movement, no appendages suited for escape, and no evidence of chemical deterrence. Their forms are broad, often flattened, and remain in fixed positions for extended periods. In a setting where consumption becomes possible, such characteristics would make them readily accessible. In many ecological systems, the introduction of predation leads to a response. Organisms develop structures or behaviors that reduce vulnerability, including protective coverings, concealment, or increased mobility. Within this record, such adaptations are not observed. Across millions of years, the forms remain consistent, without clear indication of change in response to increasing interaction. This absence raises questions about how these organisms responded to the pressures they may have encountered. Two possibilities are considered. One is that the appearance of predation occurred over a timescale shorter than the organisms could adapt to, introducing a new condition too rapidly for a response to emerge. Another is that the biological framework of these organisms did not allow for the kinds of modifications required to produce protective features. Their structure, effective under earlier conditions, may not have supported the development of defensive strategies. There are, however, aspects of the record that do not align fully with this explanation. Some of the largest and most exposed forms persist longer than others, remaining present even as changes occur. If predation were the primary driver, it would be expected that these forms would decline first, yet the pattern does not consistently reflect this. Their continued presence suggests that other factors may have influenced their persistence. Additionally, in environments where evidence of direct interaction is limited, such as deeper settings, similar patterns of disappearance are observed. The absence of clear predatory marks in these areas indicates that the process was not confined to regions where such activity is most evident. The changes appear across different environments, suggesting a broader influence. Taken together, these observations support the idea that predation played a role, contributing to shifts in how organisms interacted and survived. At the same time, the inconsistencies within the record indicate that it does not provide a complete explanation. The disappearance of these forms reflects a process influenced by multiple factors, with predation acting as one component within a larger transition. Beneath the visible forms, a quieter structure held everything in place, a living surface that functioned less like soil and more like a continuous skin stretched across the seafloor. It was not fixed in the way rock is fixed, yet it resisted disturbance, sealing what lay below and maintaining a delicate chemical balance that defined the world above it. Within this setting, entire communities depended on stability that extended downward into layers unseen. Over time, a different kind of movement began to pass through this surface. Instead of drifting across it, certain organisms pressed into it, displacing material and creating narrow passages that reached below. These motions were small at first, localized interruptions that the surrounding structure could absorb. The surface retained its continuity, closing over shallow disruptions and restoring its layered arrangement with quiet persistence. As this activity increased, the nature of the surface began to change. Each intrusion carried oxygen into spaces that had long remained isolated from it, altering chemical gradients that had been stable across immense spans of time. Layers that once remained sharply defined began to blur, their boundaries softened by repeated mixing. The integrity of the system, once maintained by stillness, started to weaken under constant disturbance. There appears to have been a threshold within this process. When disturbance remained limited, the surface could recover, re-establishing its structure faster than it was altered. Beyond a certain intensity, recovery slowed, and disruption accumulated. Each new passage added to a growing network that prevented the surface from sealing itself. The system shifted from resilience to decline, not through a single event, but through the steady accumulation of change. This transition is recorded in stone with unusual clarity. Layers that once show fine, uninterrupted laminations give way to textures that appear mottled and churned, as if the sediment had been stirred repeatedly before it hardened. This pattern appears across distant regions, suggesting a widespread transformation rather than a localized phenomenon. The boundary between these textures marks a fundamental change in how the seafloor functioned. For the organisms that relied on the earlier state, this shift removed the conditions that sustained them. Their mode of existence depended on a surface rich in dissolved organic material, maintained by the balance between microbial activity and chemical isolation. As mixing increased, that balance dissolved. Nutrients that, once accumulated within the system, dispersed more freely, reducing the concentration available at the interface where these organisms lived. There is a quiet irony in this process. The accumulation of organic material, produced over long intervals by the communities themselves and their microbial surroundings, contributed to conditions that favored the organisms capable of disturbing the surface. What had once supported one form of life gradually enabled another, one whose behavior undermined the very structure that had made it possible. This pattern reflects a broader ecological principle, where organisms alter their environment in ways that reshape the conditions of their own existence. Such transformations do not require intention, only interaction sustained over time. The environment becomes both product and constraint, evolving alongside the life it supports. Within this perspective, the disappearance of earlier forms does not arise from sudden loss, but from the gradual removal of the conditions that defined them. As the surface fragmented and its chemistry shifted, the world they depended on receded. In its place, a different kind of seafloor emerged, one no longer structured by stillness, but by motion, mixing, and continual change. Long before anything visible shifted along the seafloor, the water itself had begun to behave differently, carrying within it signals of a system adjusting on a planetary scale. These changes left no immediate shapes or impressions, yet they altered the conditions every organism depended on, quietly rewriting the rules of survival through chemistry rather than motion. Within the rock record, this transformation appears as a striking deviation known as the Schramm excursion, a period when the balance of carbon isotopes moved sharply away from its long-held range. Values of carbon-13 dropped across marine sediments worldwide, forming a signature that persists across continents. This shift suggests that an immense reservoir of carbon, lighter in isotopic composition, entered the ocean system over a relatively short geological interval. The source of this carbon is most often linked to the oxidation of dissolved organic material that had accumulated over vast spans of earlier time. For billions of years, the ocean had stored this material in deep waters, largely isolated from oxygen. As conditions changed, that reservoir may have been exposed, releasing its contents into circulation and altering the chemical structure of the ocean as a whole. Alongside this reorganization of carbon, the distribution of oxygen within the ocean remained uneven. Even as the atmosphere held more oxygen than in earlier eras, the deeper waters did not share that uniformity. Evidence from iron and trace elements indicates that zones lacking oxygen expanded and contracted across the seafloor. These anoxic regions were not constant, but their periodic presence introduced instability into environments that had once remained chemically consistent. The sulfur cycle adds another layer to this shifting system. Isotopic signatures, preserved in sediments, reveal intervals when hydrogen sulfide accumulated in bottom waters. This compound, produced by microbial processes in oxygen-poor environments, carries a toxicity that affects most complex cells. When present in sufficient concentration, it alters metabolic pathways, disrupting processes that depend on oxygen and interfering with cellular function at a fundamental level. Despite these disturbances, the record does not show an immediate disappearance of the organisms that inhabited these environments. Many persisted through these intervals, suggesting that no single chemical event acted as a sudden endpoint. Instead, the pattern reflects repeated episodes of stress, each one altering conditions without entirely removing the possibility of survival. Across time, the cumulative effect of these changes becomes more apparent. Fluctuations in carbon cycling would have influenced nutrient availability and energy flow, while oscillations in oxygen levels introduced unpredictability into habitats that depended on chemical stability. Periodic exposure to hydrogen sulfide added an additional constraint, narrowing the range of conditions under which organisms could function effectively. This combination of factors suggests a gradual reduction in ecological resilience. Systems that had once operated within narrow but stable boundaries were now subject to repeated shifts, each one requiring adjustment. Over extended intervals, the capacity to recover from these disruptions may have diminished, leaving communities more vulnerable to additional pressures emerging at the same time. Modern environments offer a partial reflection of these conditions. In certain enclosed basins, deeper waters remain depleted of oxygen and enriched in hydrogen sulfide, creating sharp chemical boundaries that limit the types of organisms able to persist there. These settings demonstrate how chemistry alone can define the structure of an ecosystem, shaping not only where life exists, but how it functions within constrained conditions. Seen in this light, the late changes in ocean chemistry form a background process rather than a singular event, a slow reshaping of the medium itself. The water that once sustained a particular way of life gradually shifted, altering its composition until the conditions that had supported that life no longer held steady. In the quiet intervals between preserved impressions, there exists another possibility, one not written directly in stone, but suggested by absence as much as presence. It rests on the idea that disappearance can sometimes be a change in form so complete that continuity becomes difficult to recognize. What seems to vanish may instead continue along a path that no longer resembles its origin. Genetic evidence, drawn from the living world rather than the fossil record, points toward lineages of animals diverging far earlier than their first clear appearances in stone. When sequences of DNA are compared across modern groups and calibrated against known geological markers, their branching patterns extend back into deep time. Many of these divisions fall within the span associated with the Ediacaran, indicating that the foundational branches of animal life had already separated long before their bodies became visible in abundance. This implies a hidden phase, a period when early animals existed in forms too small or too delicate to leave a lasting record. Their presence would not have been marked by large structures or easily preserved shapes, but by subtle movements and simple interactions with the surface beneath them. They would have lived alongside the more conspicuous organisms, sharing the same environments while remaining largely invisible to the processes that create fossils. A glimpse of this hidden world appears in the form of Icaria wariusha, a small, oval organism identified from trace evidence in ancient sediments. Its body, only a few millimeters in length, shows a clear distinction between front and back along with the capacity for directed movement. The burrows associated with it are simple, shallow paths that reflect a controlled interaction with the surface. Despite its modest size, it represents a fundamental shift, a body organized along a bilateral axis that defines the structure of most animals that exist today. Within the same interval, other organisms offer hints of continuity across the boundary. Kimberella, with its evidence of active feeding, has been proposed as an early relative of mollusks, linking it to a lineage that persists in countless forms. If this interpretation holds, then at least one recognizable thread extends from the Ediacaran into the broader tapestry of later life, carrying forward traits that would be refined over time. Yet this continuity is not universal. Many of the most distinctive forms from that earlier world leave no clear descendants. Their structures do not reappear in later records, nor do their patterns of growth find obvious parallels in the bodies that follow. If they contributed to later life, the transformation has obscured the connection beyond recognition. Their disappearance, in these cases, appears complete, leaving only impressions that resist direct comparison. The result is a mixed picture, one where survival and loss coexist. Some lineages persist, reshaped into forms that become more familiar, while others end without continuation. The boundary between these outcomes is difficult to trace, as the evidence required to distinguish them often lies in features that do not preserve well. Internal anatomy, soft tissues, and subtle developmental pathways remain largely inaccessible, leaving only partial clues to guide interpretation. In this view, the transition from one world to the next becomes less a replacement and more a reorganization. Certain elements carry forward, adapting to new conditions and becoming part of a different ecological structure. Others fade entirely, their roles unfilled by direct descendants. What remains is a continuity that is both real and incomplete, linking distant past and present through pathways that can be glimpsed but not fully reconstructed. There are traces that do not take the shape of bodies, residues that persist long after form has vanished, carrying information in patterns too small to see. Within certain rocks, fragments of ancient molecules remain embedded, altered but still recognizable, offering a different kind of record. They do not show outlines or impressions, yet they preserve something more intimate, the chemistry of life itself. Among these approaches is paleoproteomics, a field that attempts to recover ancient proteins from fossil material. Proteins, though fragile in living systems, can under rare conditions endure far longer than genetic material. Their sequences, when preserved, retain signatures of biological identity, linking them to particular groups of organisms through shared structural patterns. In younger fossils, these methods have already revealed relationships that anatomy alone could not resolve. Extending this approach into deeper time suggests a possibility. If proteins could be extracted from the oldest preserved organisms, their sequences might be compared directly with those of living life. Such a comparison could anchor uncertain forms within the broader tree of biology, not through appearance, but through molecular continuity. In theory, this would provide a direct answer to questions that morphology leaves unresolved. In practice, this remains beyond current reach. The conditions required to preserve proteins across more than 500 million years are exceptionally restrictive. Heat, pressure, and chemical alteration steadily degrade these molecules, breaking them into fragments that lose their original structure. Despite this, researchers continue to examine the most promising sites, searching for environments where preservation may have occurred under unusually stable conditions. One such environment is found in the phosphate deposits of South China, where microscopic organisms have been preserved in remarkable detail. These formations capture cellular structures in 3 dimensions, revealing boundaries and arrangements that resemble living tissues. Within these rocks, clusters of dividing cells have been discovered, their organization suggesting early stages of development. These formations sparked a debate that remains unresolved. Some interpretations view them as early animal embryos, their division patterns echoing those seen in modern organisms. If correct, this would place the origins of complex development earlier than previously confirmed. Other interpretations suggest a different identity, pointing toward large sulfur bacteria or resting stages of algae. The structures alone cannot settle the question, as their external form can be explained by multiple biological pathways. Beyond proteins, another class of molecules offers a more durable record. Lipids, particularly those forming cell membranes, can survive extensive geological alteration. When preserved, they transform into compounds known as steranes, which retain the chemical signatures of their original sources. These molecules provide a way to trace the presence of complex cells independent of visible fossils. The discovery of cholesterol derivatives within certain ancient specimens has already demonstrated the power of this approach. Cholesterol is associated with animal cell membranes, and its presence indicates a biochemical affinity that morphology alone could not confirm. Expanding this method across different sites allows researchers to map the distribution of such signatures through time, revealing when and where different types of life emerged. Yet even this molecular perspective has limits. Chemical identity can indicate broad categories, distinguishing between major groups such as animals, fungi, or bacteria, but it does not reveal the form those organisms took. A biochemical signal may identify an organism as belonging to a particular lineage, while leaving its structure, behavior, and ecological role entirely unknown. In this way, molecular evidence adds depth without completing the picture. It offers a parallel narrative, one written in chemistry rather than shape, extending understanding into places where fossils alone cannot reach. Together, these traces suggest that the history of life is recorded in multiple layers, each revealing different aspects of a past that cannot be seen all at once. The pace of discovery has begun to quicken, as if a long, quiet archive is being opened drawer by drawer, each layer yielding forms that were once overlooked or misunderstood. In recent decades, the number of described organisms has grown rapidly, surpassing what earlier generations had assembled across far longer spans of time. What once seemed sparse now appears crowded with variation, each edition reshaping the outline of that distant world. Among these finds is Aurora lumina attenboroughii, a form uncovered in the same English landscape that first challenged earlier assumptions. Its structure rises in a narrow column before dividing into a forked upper portion, a shape that suggests a division of function within a single body. Dating to around 560 million years ago, it occupies a position near the closing phases of that interval, yet its organization hints at patterns that become more familiar later. Its exact relationship remains uncertain, but its presence expands the range of possibilities for how bodies were arranged during that time. At sites where entire communities were preserved in place, new methods have begun to reveal details that were once hidden within the rock. Photogrammetry and 3-dimensional modeling allow surfaces to be reconstructed with extraordinary precision, capturing subtle variations in relief that the eye alone cannot resolve. When applied to deep water assemblages, These techniques have shown that the branching forms extend inward as well as outward, following repeated geometric patterns through multiple layers of structure. What once appeared as flat impressions now suggests a deeper organization, one governed by consistent internal rules. The geographic spread of these organisms has also widened as new sites are identified across regions once thought unlikely to preserve them. Discoveries in Mongolia, the northern reaches of Norway, and parts of Iran extend the known distribution across a broad range of ancient environments. This expanded map suggests a level of uniformity across the planet, implying that the conditions supporting these forms were not confined to isolated basins, but were maintained across connected ocean systems. Advances in imaging now allow researchers to look inside specimens without altering them. Synchrotron-based techniques pass focused beams through the rock, detecting variations in density that reveal internal structures at fine scales. In specimens that once appeared featureless, these methods have uncovered layered arrangements and subtle internal divisions, adding new dimensions to forms previously known only from their outer surfaces. In southern China, ongoing work within the Denjiang Formation has produced organisms bearing early signs of mineralized structures. These discoveries suggest that the development of rigid components may have begun earlier than once thought, appearing in multiple lineages rather than emerging from a single origin. This pushes back the timing of a transition that would later become widespread, indicating a more gradual onset of structural innovation. At the same time, computational approaches are being applied to large collections of fossil images. Machine learning systems analyze patterns across thousands of specimens, identifying variations that may not be immediately apparent to human observers. Through this process, some forms once grouped together are being separated, while others reveal connections that had gone unnoticed. The classification of these organisms continues to shift as new patterns emerge from this broader analysis. Taken together, these developments point toward a future in which multiple lines of evidence converge. Imaging, chemistry, and computation each contribute different perspectives, gradually filling gaps left by any single method. As these approaches are applied to the most carefully preserved specimens, the distinctions between categories may become clearer, not through a single discovery, but through the steady alignment of many. What passes from one world into another is not always what draws the eye. Large forms can vanish, while smaller, quieter lineages continue, carrying forward structures that prove more adaptable than those that once dominated the surface. At the boundary between these intervals, survival belonged not to the most visible, but to the most flexible. Among those that endured were organisms of remarkable simplicity. Sponges, with their porous bodies and constant flow of water through internal channels, required little in terms of structural complexity. They lacked organized tissues, lacked centralized control, and yet maintained a steady exchange with their surroundings. This simplicity became an advantage. Their mode of feeding, drawing suspended particles from the water, remained effective across changing conditions, allowing them to persist with little need for alteration. Alongside them were early members of another lineage, ancestors of forms that would later include drifting and anchored bodies alike. These organisms possessed basic tissues and simple arrangements of cells, capable of responding to their environment in ways more coordinated than earlier life. Their origins extend deep into earlier time, and as conditions shifted, they did not disappear. Instead, they expanded, diversifying into a wide range of forms that would occupy new ecological roles. Less conspicuous, but equally important, were small organisms organized along a front-to-back axis. Their bodies were soft and modest in size, leaving only subtle traces of their movement through sediment. These early bilaterians introduced a different kind of interaction with their surroundings, one based on directed motion and internal organization. Their presence across the transition provided a foundation upon which later complexity could build, as their descendants diversified into numerous forms. The continuity of life across this boundary was not limited to organisms alone. The environment itself carried forward a legacy shaped over immense spans of time. The composition of the ocean, its dissolved nutrients and chemical balances, had been influenced by earlier ecosystems. These conditions did not reset at the boundary, but persisted, forming the medium in which later life would expand. Within this continuity, genetic systems played a central role. The regulatory networks that guide the development of bodies, including sets of genes responsible for organizing structure along an axis, had already formed earlier. These systems did not originate suddenly, but were assembled gradually, existing before they were widely expressed in complex forms. When conditions allowed, these genetic tools were used in new ways, producing a diversity of structures from a shared foundation. Even as the surface environment changed, remnants of earlier systems continued to influence what followed. Organic material, accumulated over long intervals, remained within the sediment, gradually becoming available as conditions shifted. This stored energy contributed to the productivity of emerging ecosystems, supporting organisms that interacted with their environment in new ways. The appearance of a sharp boundary in the fossil record reflects a change in what is preserved as much as a change in what exists. Forms that were once common no longer appear, while others become more visible due to their structures and behaviors. Beneath this apparent divide lies a continuity of processes, lineages, and environments that link the two intervals. What emerged after this transition did not arise from absence, but from persistence. The organisms that endured carried forward essential traits, interacting with a world shaped by those that came before. In this way, the transition represents both an ending and a continuation, a shift in form built upon an underlying continuity that remained in place. Numbers alone drift without weight until they are given a place to stand. 575 million years rests easily on a page, yet it resists any instinct for proportion, slipping past the boundaries of ordinary experience. To approach it, the mind reaches for translation, for a shape that can hold its quiet vastness. Imagine the entire history of Earth drawn into a single day. Midnight marks the beginning, the first formation of a world still gathering itself. Hours pass in slow procession, each one carrying spans of time longer than anything familiar. Continents assemble and separate in silence. Oceans deepen, atmospheres shift, all within this long unfolding arc. Only near the final moments does something different begin to stir. At approximately 11:04 in the evening, the first large and complex forms appear, arriving late in a day already nearly complete. Everything that feels immediate, every recorded memory of human presence, occupies a space so brief it barely registers. The rise and fall of civilizations, the shaping of language, the accumulation of knowledge, all settle into the final 2 seconds before midnight. The rest of the day stretches behind them, vast and mostly unmarked by anything resembling the present. To step back into that earlier hour is to find a world arranged in unfamiliar ways. Land masses lay in positions that would not be recognized now, clustered and drifting under a sky that held no memory of what would follow. The ocean that would one day divide continents had not yet opened. Mountain ranges that define entire regions of the present were still far in the future, their formations separated by spans of time longer than the age of many modern lineages. Beyond the Earth, the same scale unfolds in another direction. Light travels across space at a constant pace, carrying with it a record of distant events. A beam of light that left a galaxy 575 million light years away begins its journey while those early forms moved across ancient seafloors. That same light arrives now, entering telescopes and eyes, bridging two moments that feel distant, yet are nearly adjacent on a universal scale. Closer to home, the material of the present carries its own quiet history. The atoms that compose a living body have passed through countless transformations, cycling through air, water, and living tissue over immense spans of time. Each element has been part of systems long vanished, incorporated, and released again and again, moving through different forms before settling, for a moment, into something current. Even the surfaces those early organisms once occupied remain, though altered beyond recognition. Layers of sediment have compressed into stone, preserving faint impressions of forms that once rested there. These surfaces lie embedded within landscapes far removed from their original setting, lifted, tilted, and weathered across ages. There is no direct experience that can contain such duration. It extends beyond memory, beyond analogy, beyond any measure rooted in a single life. Yet, in that distance, a certain clarity emerges. The present becomes a narrow edge along a much larger continuum, and the events that seem fixed take on a different proportion. The effort to hold this scale, even imperfectly, becomes its own way of understanding, a quiet acknowledgement of how much has unfolded before this moment and how briefly it is held. What remains of that distant world arrives as absence shaped into form. Stone holds impressions where bodies once rested, outlines pressed into sediment that hardened long after the organisms themselves were gone. The record appears detailed at first glance, yet it is built from rare moments when conditions aligned with unusual precision. Soft tissue rarely endures. For an organism to leave a lasting mark, it must be covered quickly by fine sediment, sealed away from disturbance, and held within a chemical environment that slows decay. These circumstances are not common. Across nearly 100 million years, they occurred only in scattered places, brief windows where preservation became possible. The surfaces uncovered today represent those windows alone, a handful of preserved scenes drawn from an immense span of unrecorded time. From this narrow archive, an entire living system is reconstructed. Each fossil surface offers a snapshot, a single arrangement of bodies at a particular moment. Between these snapshots lie intervals that extend for millions of years, unrepresented and silent. The continuity of life across those gaps is inferred rather than observed, a sequence imagined from isolated frames. Movement leaves traces, faint pathways etched into ancient substrates. Some organisms shifted across the surface, leaving behind patterns that suggest direction and repetition. These marks imply activity, a form of interaction with the environment, yet they reveal little about the nature of that activity. The presence of motion does not disclose whether it was guided by sensation, nor whether there existed any internal experience associated with it. The distinction between mechanical response and something more complex remains beyond reach. Reproduction, the process that sustained these communities, is largely hidden. Most known forms appear only as mature impressions, fully formed and resting in place. There are no clear sequences of development, no preserved transitions from early stages to later ones. Without eggs, larvae, or intermediate forms, the means by which these organisms produced new individuals remains uncertain. Growth can be traced in some cases through size variation, yet the full cycle of life is rarely visible. The internal structure of these organisms is also obscured. Fossils preserve surfaces, not the systems beneath them. Organs, tissues, and cellular arrangements are either lost or never recorded in the first place. Classification, which in modern biology relies heavily on internal features, must instead depend on external patterns alone. Shapes, symmetries, and surface textures become the primary guides, even when they offer only partial information. Time itself introduces another layer of distance. The period in which these organisms lived extends far beyond the span of more recent events, yet it is sampled unevenly. A small number of horizons provide detailed records, while vast stretches between them remain unobserved. Each known layer becomes a point of reference, and everything beyond those points must be bridged through careful interpretation. Within these limits, knowledge advances with caution. Certain patterns emerge with clarity: global distribution, recurring forms, ecological organization across multiple sites. Other aspects remain unresolved, held just beyond the reach of available evidence. The nature of these organisms, their precise relationships, and the processes that shaped their history continue to resist complete definition. The record does not offer a complete narrative. It provides fragments, preserved under specific conditions, separated by intervals that cannot be directly examined. From these fragments, a picture is assembled, provisional and open to revision. The silence between the preserved moments remains, a reminder that even with refined tools and careful analysis, some parts of this distant chapter will remain unspoken, held within the stone that carries only what it could keep. Before any lineage could be named or compared, there was simply presence, bodies arranged across the seafloor, participating in processes that extended far beyond their individual forms. For tens of millions of years, these organisms did not wait for what would come next. They lived within a system they were actively shaping, altering the chemistry and structure of their environment through the quiet accumulation of daily activity. Organic matter moved continuously through this world. Material produced at the surface settled downward, meeting communities that absorbed, transformed, and redistributed it. Each organism contributed to this flow, either by taking in dissolved compounds or by processing the thin layers beneath them. Over time, these interactions influenced the balance of carbon in the ocean, shifting how it was stored, released, and cycled through different reservoirs. The cumulative effect extended beyond any single surface, gradually altering the composition of the surrounding water. As organic material was consumed and broken down, oxygen played an increasingly central role. The shallow seas where many of these organisms lived became zones of active exchange, where metabolic processes and microbial activity together influenced oxygen availability. The presence of these communities contributed to maintaining conditions that could support more demanding forms of metabolism. The oxygen that would later sustain more complex animals was not introduced suddenly. It was shaped over long intervals by systems already in motion. Beneath these communities, sediment gathered slowly, layer upon layer. Each generation left behind traces of organic matter that became incorporated into the accumulating substrate. Over immense spans of time, this material was buried, compacted, and preserved. These deposits formed a record not only of individual organisms, but of the processes that sustained them. In later ages, this stored carbon would become part of new cycles, feeding organisms that depended on richer energy sources, and in some cases, transforming into the geological materials studied in the present. Within this environment, patterns of organization emerged. Different forms occupied distinct positions, some remaining close to the surface, while others extended upward into the surrounding water. Space was not used uniformly. It was structured, with areas of higher density and zones of separation, suggesting a balance between growth, resource access, and the limits imposed by neighboring organisms. These arrangements represent early expressions of ecological organization, a distribution of roles that would later become more intricate. Interactions also began to introduce pressure into the system. Some organisms moved across the surface in ways that altered the substrate itself, disturbing the layers that had previously remained stable. These movements created new conditions, modifying the environment in ways that affected other inhabitants. The relationship between organism and environment became increasingly dynamic, with changes in one influencing the possibilities available to others. Over time, these interactions accumulated into broader transformations. The seafloor was no longer a passive surface, but an active interface where biological and chemical processes were tightly linked. Each organism, through its existence, contributed to a shared system that extended across regions and persisted through successive generations. This long interval of activity established conditions that would carry forward. The chemistry of the ocean, the distribution of organic matter, and the emerging patterns of interaction all formed part of a foundation that later developments would build upon. The changes were not abrupt. They unfolded gradually, shaped by countless small contributions that, taken together, altered the trajectory of life. In this way, the significance of these organisms lies not only in their forms, but in what they sustained. Their presence influenced the pathways available to future systems, embedding within the environment a legacy of altered conditions and accumulated potential. Far from any shoreline or fossil surface, the same story extends outward into space, carried as a quiet comparison between one world and many unseen others. The record left behind here becomes more than a history. It becomes a reference, a single example of how life might unfold when given time and the right sequence of conditions. For most of this planet's existence, life remained small and self-contained, thriving at scales that rarely leave visible traces. Nearly 4 billion years passed in this state, with vast microbial systems shaping oceans and atmosphere without producing bodies that rise above the surface. If this long interval is not unusual, then countless worlds may follow a similar path, hosting abundant life that never assembles into larger forms. Complexity, in this view, is not the default outcome, but a late and uncertain development. The transition required more than simple persistence. It depended on a convergence of factors, each unfolding on its own timeline. Episodes of global freezing altered the distribution of nutrients, while shifts in atmospheric composition gradually increased the availability of oxygen. Ocean chemistry changed in ways that supported new forms of metabolism. At the cellular level, a rare union between different kinds of organisms produced the internal complexity necessary for larger structures. Each step alone might not be sufficient. Together, they created a narrow window in which new possibilities could emerge. Beyond Earth, there are places where some of these ingredients appear to exist. Beneath the icy surfaces of Europa and Enceladus, subsurface oceans persist, warmed by internal energy and enriched with chemical gradients that can sustain life. If organisms are present there, they may resemble the earliest stages seen here, microbial, widespread, and enduring. Yet without exposure to surface processes that alter atmospheric composition, and without cycles that redistribute nutrients on a planetary scale, the pathway toward larger forms may remain closed. This pattern suggests that while life itself might arise under a variety of conditions, the progression toward complex structures could be far less common. A planet may sustain living systems for billions of years without ever crossing the thresholds required for multicellular organization. Even when those thresholds are approached, the alignment of necessary factors may be brief, contingent, and easily disrupted. At the same time, the forms that complexity takes need not follow familiar patterns. The organisms preserved from this interval demonstrate that large, organized bodies can exist without resembling later animals or plants. Their structures, symmetries, and modes of existence broaden the range of what complexity can look like. If similar processes occur elsewhere, the resulting life may not align with expectations shaped by modern ecosystems. Recognition depends on understanding that complexity is defined by organization and interaction, not by resemblance to known forms. This perspective also touches on a larger silence. The absence of detectable signals from distant systems does not require the absence of life. It may reflect the rarity of transitions that lead from simple organisms to technological expression. Each step in that progression carries its own requirements, and the combination of all steps may be uncommon even on worlds where life is otherwise successful. The history recorded here offers both expansion and restraint. It widens the range of possibilities for how life can exist, while also narrowing the conditions under which it may advance. The presence of complexity becomes something contingent, shaped by timing and circumstance rather than inevitability. In this way, the ancient seafloors reflect outward, suggesting that the universe may hold many living systems that never reach beyond their earliest forms, and that those which do are shaped by a sequence of events that need not repeat. A pattern appears when the record is followed to its edge, a pattern that does not continue into the forms that come later. Some of the most intricate bodies vanish without clear descendants, their designs ending where the stone record fades. This absence invites a quiet question about what it means for a lineage to persist or to end. Certain structures seem to belong entirely to that interval. Fractal forms rising from the seafloor, bodies organized in symmetries that do not recur, surfaces divided into repeating compartments with no modern equivalent. If none of these contributed directly to later lineages, then their history stands apart, a long interval of biological activity that did not extend forward in recognizable form. By a narrow definition centered on ancestry, this would appear as failure, an elaborate system that left no trace in what followed. Yet, the path of evolution is not fixed in advance. The sequence that led from those early communities to later animals depends on a series of conditions aligning in a particular way. A slight variation in environment, timing, or interaction could have shifted the outcome. The forms that dominate later periods were not guaranteed. They emerged within a specific set of circumstances, and different conditions might have favored entirely different structures. In this sense, what survives is not necessarily what was most capable, but what happened to align with the unfolding context. Even without direct descendants, the influence of these organisms extended into the systems that followed. Their presence altered the chemistry of the ocean, contributed to the accumulation of organic material, and shaped the physical structure of the seafloor. These changes persisted beyond the disappearance of the organisms themselves, forming part of the environment in which later life developed. The impact of a lineage can remain even when its form does not. This raises a deeper difficulty in how success is defined. If success is measured only by the continuation of a lineage, then any organism without descendants is considered incomplete. Yet, A different perspective considers duration, distribution, and ecological integration. A community that occupies global environments for tens of millions of years, maintaining stable populations and interacting within a complex system, demonstrates a form of success that does not depend on continuation beyond its own time. There is also evidence that complexity arises more than once. Multicellular organization has appeared independently in multiple branches of life, emerging under different conditions and producing distinct forms. This suggests that the transition from simple to complex structures is not a singular event, but a recurring possibility. The organisms of this interval may represent one such emergence, a version of complexity shaped by its own context, rather than a direct precursor to later forms. Seen in this way, the idea of a dead end becomes less certain. The absence of descendants does not erase the significance of what existed. Each lineage occupies its own span, contributing to the broader system in ways that may not be visible through ancestry alone. The value of that existence lies in its participation in the processes that shape the world at that time. The question then shifts from outcome to presence. Instead of asking whether these organisms led to something else, it becomes possible to consider what they were while they existed. Their structures, interactions, and duration form a complete chapter, one that does not require continuation to hold meaning. In the quiet space left by their disappearance, what remains is not simply an ending, but a recognition that existence can be significant within its own boundaries, independent of what follows beyond it. There is no single horizon that marks the end, no layer thick with ash, no sudden interruption that separates one world cleanly from the next. Instead, the record shifts gradually, almost imperceptibly at first. Forms that once filled entire surfaces become less frequent, their outlines thinning across successive layers until they are no longer present. What replaces them does not arrive in a single wave. It accumulates, spreading through the same spaces until the earlier patterns can no longer persist. This kind of disappearance carries a different character from more abrupt endings. There is no external force imposed from beyond the system, no singular event that can be pointed to as the cause. The change arises from within, driven by processes already underway. New forms alter the environment in small ways, and those alterations, repeated across time, reshape the conditions that once supported the earlier inhabitants. The earlier communities had developed within a framework that remained consistent for long intervals. Their structures, interactions, and modes of existence aligned with that stability. Over tens of millions of years, this alignment became precise. Each organism fit its environment closely, adapted to conditions that did not require rapid change or defensive strategies. In such a setting, efficiency and persistence reinforced one another, producing systems that were both widespread and enduring. When conditions began to shift, that same precision became a limitation. Adaptations that had been advantageous within a stable context offered little flexibility outside it. The environment did not transform suddenly. It changed through the cumulative effects of new activities, subtle alterations in the seafloor, the redistribution of resources, and the increasing complexity of interactions. These changes did not resemble a threat in the conventional sense. They were part of the same system, emerging from processes that had been present all along. The transition reveals a broader pattern. Systems that develop under stable conditions often refine themselves toward that stability. Their structures become optimized for a particular range of variation, narrowing the pathways through which they can respond to change. When the environment moves beyond that range, The very features that once supported persistence can limit the capacity to adjust. The earlier world had no need to anticipate disruption. Its longevity reinforced the expectation that conditions would continue as they had. This expectation was not a conscious one, but it was embedded in the forms and interactions that defined the system. As new processes accumulated, the environment shifted into a state that no longer supported those forms. The change did not target them directly. It altered the framework within which they existed. The forms that replaced them were not introduced as external agents. They emerged from the same continuum, shaped by conditions that had been developing over long periods. Their presence introduced new interactions, new ways of modifying the environment, and new structures that could persist under the changing conditions. In doing so, they extended the system into a different configuration, one that no longer included the earlier forms. The absence that follows is not empty. It is filled with a different arrangement, one that carries forward elements of what came before, while leaving others behind. The earlier world remains part of the sequence, its influence embedded in the conditions that allowed the transition to occur. In this gradual replacement, a principle becomes visible. Persistence is not guaranteed by duration alone. Systems can endure for immense spans and still give way when the context that sustains them shifts. The processes that produce stability also carry within them the potential for change, and that change unfolds without a clear boundary between continuation and ending. Beneath a shallow sea that no longer exists, within sediment that would one day harden into stone, a small form pressed forward through grains too fine to hold shape. Its movement left only a faint disturbance, a narrow trace that would vanish almost as quickly as it formed. It carried within its body a simple architecture, a front that encountered the world first, a rear that released what had been processed, and a continuous passage between them. Nothing in its surroundings marked it as significant. It was one presence among many, moving through a landscape already shaped by earlier lives. Within that small body, sequences of molecules folded and unfolded, directing growth, division, and repair. These sequences, arranged along strands coiled within each cell, encoded patterns that could be copied, altered slightly, and passed forward. They did not anticipate what would come. They operated within the immediate demands of existence, maintaining structure, guiding development, allowing the organism to persist from one moment to the next. Across vast intervals, those sequences continued, carried by descendants that did not resemble their origin. Forms changed gradually, sometimes in ways that left little visible trace, sometimes in ways that reshaped entire bodies. Fins emerged, then limbs capable of bearing weight beyond water. Structures adapted for sensing light, detecting chemical gradients, coordinating movement across greater distances. Each transformation remained connected to what preceded it, not as a sudden departure, but as an extension of inherited instructions responding to new conditions. The continuity is not visible in any single moment. It becomes apparent only when viewed across the long arc, where the same underlying framework persists through shifting expressions. A body capable of processing nutrients through a single passage becomes, over time, the basis for more complex systems. Coordination between cells develops into networks that transmit signals. Simple responses to stimuli become more elaborate forms of interaction with the surrounding environment. At no point does the sequence pause. Each generation follows another, uninterrupted, carrying forward what was preserved and introducing small variations that may or may not persist. The path is neither directed nor random in any simple sense. It unfolds through the interplay of inheritance and circumstance, shaped by conditions that favor some configurations over others. The forms that shared the earlier seafloor existed alongside this lineage without intersecting it directly. Their structures followed different patterns, their modes of existence aligned with conditions that did not require movement or internal processing in the same way. They occupied the same environment, yet their trajectories did not converge. The lineage that continued did so quietly, without altering the broader landscape in ways that would have been immediately apparent. Over time, that quiet continuity produced organisms capable of perceiving their surroundings with increasing detail, of coordinating actions across larger bodies, of storing and processing information in ways that extended beyond immediate stimuli. These capacities emerged gradually, each step grounded in the same molecular mechanisms that had operated in the earliest forms. The thread that connects those distant moments to the present is not a single line, but a vast network of continuities, each branch linked through shared ancestry. The structures within a modern body, the pathways that guide development, the molecules that sustain life, all carry traces of that origin. They have been modified, recombined, and refined, yet they remain built upon the same foundational patterns. In this way, the passage from that small organism in ancient sediment to the present moment is not a sequence of separate events, but a continuous unfolding. The distance between them is measured not only in time, but in transformation. Each stage preserving something of what came before, while extending it into new forms. Across a floor of fine sediment, a quiet arrangement persists, held in a stillness that belongs to another order of time. Nothing moves quickly here. Forms rise gently from the surface, some spreading low, others extending upward into water that carries no light. There is no disturbance beyond the faint drift of particles, no sudden change in direction, only a slow exchange between body and surrounding medium. Sound, as later life would come to use it, has no presence. No vibrations carry intention. No signals pass from one organism to another in pulses or calls. Instead, interaction unfolds through gradients of chemistry, invisible shifts in concentration that move across the seafloor like currents without edges. Each organism participates in this exchange, taking in dissolved matter, releasing byproducts, altering the immediate environment in ways that ripple outward without urgency. In deeper regions, far below where sunlight could ever reach, structures extend into darkness with precise repetition. Their geometry unfolds in branching patterns that echo themselves at different scales, each division mirroring the last. Growth here is not directed toward illumination, but toward opportunity, a quiet optimization that gathers what the water offers. The surrounding space remains unchanged by brightness or shadow, yet these forms persist, anchored and steady, drawing sustenance from what cannot be seen. Beneath them lies a continuous surface, a living layer that predates their arrival by spans too long to measure easily. It binds the sediment, holds it in place, and provides the medium through which nutrients are stored and transformed. This surface is neither passive nor fixed. It responds, repairs, and reshapes itself over time, maintaining a stability that supports everything above it. Even as new forms emerge and occupy its expanse, it remains the foundation upon which their existence depends. Moments arrive when this quiet continuity is interrupted. Fine material settles rapidly, covering entire communities in a single event. Where once there was slow exchange, there is now preservation. Shapes are captured as they were, their positions fixed, their outlines held with a precision that endures beyond their living state. What remains is not the organism itself, but the record of its presence, an imprint that carries the form forward while the substance is gone. Elsewhere, surfaces bear different traces, subtle depressions where movement once occurred. Patterns of interaction are recorded not as bodies but as the absence they created, spaces where material was displaced, where feeding altered the texture of the ground. These impressions persist as negative forms, evidence of activity that no longer exists, yet remains legible in the structure of the rock. Across distant regions, layers of stone hold these records in quiet succession. Some remain exposed, open to the present, while others lie buried, waiting without urgency. The passage of time does not erase them completely. It transforms their context, shifts their position, carries them across changing landscapes until they appear again, accessible to those who encounter them. What endures is not the life itself, but the trace it left behind, a pattern preserved through processes that outlast the organisms that created it. These impressions are fragments of a world that no longer operates under the same conditions, yet they remain connected to everything that followed. The continuity is not in the forms, but in the processes that allowed those forms to exist and be recorded. Within that continuity, the present moment takes its place, shaped by sequences that extend far beyond immediate perception. The capacity to observe, to reflect, to recognize patterns in stone arises from the same unfolding that produced those early imprints. The distance between them is vast, yet it is bridged by an unbroken sequence of change. In this way, the quiet seafloor and the present awareness belong to the same continuum, different expressions of processes that began long before either could be recognized.